exciting molecular biology and molecular biology technique. These are methods scientists use to study the tiny building blocks of our life, DNA, RNA, and proteins. Whether you are new to this field or just curious, I'm here to break it down in a simple and understandable way. Um, okay, let's get started our video. First of all, we move to the next slide. All right. So that is the molecular biology and molecular biology techniques. First of all, I have put the molecular biology definition. I'm sure you have gone through this. Molecular biology is study of biology at the molecular level, right? So we are going to study at the molecular level. Molecular biology, this field overlaps with other areas, biology and chemistry, particularly genetics and biochemistry. Molecular biology chiefly concern itself with understanding the interaction between the various systems of a cell, including the interrelationship of DNA, RNA, and protein synthesis, and learning how these interactions are regulated. So actually speaking, if we study the DNA of the cell, that is genome. Okay, genome means all the DNA inside the plant or animal cells nucleus. Okay, if we have chloroplast inside the DNA called chloroplast genome. If we have a mitochondrion, its DNA also called mitochondrial genome, right? So the genome is all the DNA. Then, if you're going to look the RNA of a cell, RNA of the cell, it is a transcriptome. Because the transcribed DNA are the RNA, RNA is ribonucleic acid. No? Okay, and so not all genes are transcribed, right? About 2 or 3% of the gene inside the cell is transcribed, particular time, right? And if you're going to study about the protein, protein or other ones actually does not function. Proteins are other ones actually the function of the cell, right? If it is photosynthesis, enzyme reaction, whatever, everything inside the cell doing the protein or inside like this. It is the protein most of the time. RNA also has some function, but protein are other ones that do function of the whole cells. If you're going to study all the protein of the cell called proteomes, okay, once mean everything's inside the cell doing by proteins. We are studying the protein, it's called proteome, right? So now we move to the next slide. As I said, techniques of molecular biology involve isolation of DNA, electrophoresis of DNA, hybridization and blotting techniques, technique for DNA amplification, restriction, fragment length polymorphisms, DNA fingerprinting, etc., etc. Thousands of techniques used in molecular biology. I put this slide some popular and more using in the world. in this field. It's some of very small amount of these techniques. There are many thousand techniques. No? I put in this slide is very little amount of the techniques. Right. Mm. Okay, I will explain each of these DNA techniques with examples from everyday life. Isolation of DNA. This is like taking a piece of uh, string, string in DNA out of the messy dough. Dough means cells. So you can look at it closely. For example, scientists might isolate DNA from a crime scene to identify who was there, right? That's the isolation of DNA. Next one is electrophoresis of DNA. Um, imagine you have a bunch of different size beads and you push them through a jelly. The smaller beads move fast and the bigger ones, the smaller beads move faster than the bigger one, right? Electrophoresis is a way to separate DNA pieces by size using electric fragment. Uh, Electrophoresis is a way to separate DNA pieces by uh, size using the electric current. It's like finding out who can run faster in the race. Understood? Uh, Next, we move to the next topic. Okay, hybridizing and blotting techniques. This is like matching socks. If you have a peel of mixed socks, you can use the matching patterns. Patterns mean probe to find pairs. In DNA, a probe can find and stick to specific DNA sequence. This helps in detecting, creating certain genes, right? Uh, this is the uh, hybridization and blotting technique. Techniques for DNA amplification. Next, fourth one is technique for DNA amplification. Imagine you have a book with only one page of a story. You have a book with only one page of a story, right? You need more copies to share with your friends and others, right? DNA amplification, like making photocopies certain, um, uh, creates many copies of a specific DNA fragment. This helps scientists to study it more easily, right? Understood what is the techniques for the DNA amplification? Um, Now you have an idea what is DNA amplification. Then we move to the next topic. Next one is um, restriction fragment length polymorphism. It's mean RFLP. This is like cutting a long piece of string of a specific place and seeing how the pieces differ in length. RFLP use special scissor, special scissor mean enzyme. It's an enzyme. I will explain it. I will explain it another video from another video, and then I will this explain now very just explain what it is. Okay. Um, scissor like enzyme. It's an enzyme. Um, to cut DNA at certain spots. And the pattern of the pieces can tell us about genetic differences like in paternity test. Last one is DNA fingerprinting. This is like using fingerprint to identify a person. Everyone has unique DNA, everyone has unique DNA and DNA fingerprints. Look at a specific part of DNA to tell people apart. This is used to forensic science to solve crime or in genealogy, genealogy to find about family history. Understood? That's a thing. That is the DNA fingerprint. Next, we move to our next slide. This is uses and importance of molecular biology techniques. Which I'll explain it. Give an examples. Disease diagnosis. First one is disease diagnosis. Doctor can use molecular biology techniques to find out what's making someone sick. For example, they can detect virus like COVID-19 in a person's body by looking for virus DNA or RNA. Next one is DNA profiling or forensics. Uh, when police find evidence at a crime scene, they can use DNA profiling to find out who was there. It's like finding a fingerprint, but using DNA to match their evidence to a person. Next one is PCR, polymerase chain reaction. This technique makes many copies of a specific DNA fragment, like photocopying a page from a book. It's used to many areas like testing for infection for studying genes. Paternity test. This used to find out who a child's biological father is. Scientists compare to child's DNA with potential to father's DNA. This helps to confirm family relationship. RFLP I mentioned earlier, restriction fragment length polymorphism. Scientists use this technique to cut DNA into pieces and see how they differ in length. This can help in studying genetic differences and relationship, like identify a person or plant variety. Cloning genes. Cloning gene means making exact copying of a specific gene. Scientists do this to study the gene functions or produce useful protein like insulin for diabetes treatments. And next one is GMO, genetically modified organisms. Scientists can change the DNA of plants or animals to give them new traits. For example, some crops are modified to uh, resist pests and help farmers to grow more foods. Last one is DNA sequencing. DNA sequencing reads the exact code of the DNA letter. A, T, C, G. This is helps scientists to understand genes and how they affect health and behavior. Example, it can help identify genetic disorders or personalize medical treatments. Right? I think all these are understood. Molecular biology techniques are the backbone of modern biology and medicine. They help us understand, diagnose, and even treat disease. If you enjoy this overview, make sure to like this video and subscribe to MicrobioTech Insight. For more deep dive into the world of microbiology and biotechnology, um, got any question or topics you would like to cover? You would like me to cover? Drop the comment below. Thanks for watching and see you next video.